Hi everyone. This video will summarize the 6 outcomes of the antigen-antibody reaction. The first outcome when the antigen binds to the antibody is the activation of the B lymphocyte. Antibody like the IgD class that occurs on the surface of the B cell can always act as the receptor for the B cell to recognize the antigen on the foreign body or the pathogen. The binding of this antigen to the antibody on the surface of B cell will activate the B cell and thus enable the B cell to undergo the clonal selection. When the activated B cell is being exposed to the cytokine, then the B cell will undergo clonal selection to produce the memory B cell and also the effector cell called plasma cell. Then later on, this plasma cell will release the antibody to attack or eliminate this antigen. Next, the second outcome is the opsonization and phagocytosis. Antibody can use their FAB region to bind to the antigen, and at the same time, they can also use their FC region to bind to any FC receptor on the surface of our cell. Normally, phagocyte will have the FC receptor on their surface. So this antibody, after they bind to the antigen with the FAB region, then they will bring this antigen towards the phagocyte and bind to the receptor on the surface of the phagocyte through the FC region. So meaning that the antibody will bind the antigen and bring and cause this pathogen to stuck at the surface of the phagocyte by binding their FC region to the FC receptor on the surface of phagocyte. So when all of these pathogens are stuck on the surface of this phagocyte, the phagocyte can then easily do the phagocytosis and engulf all of these pathogens. So the antibody in this case is acting as the opsonin. It's acting as the opsonin to cause the bacteria or the pathogen to be phagocytized. So this process is the so-called opsonization. IgG is normally a very strong opsonin, while IgA is a weaker one. The third outcome is the antigen clumping and inactivation of the toxin. Antibody class like the IgG, IgM, and IgA can use the neutralization or agglutination or precipitation in order to protect our body against the antigen. Neutralization means that the antibody go and bind to the antigen and coat the whole pathogen. So when the pathogen is being coated like this, then that means that this pathogen is no longer able to bind to our cell and cause any harmful effect to our cell. So with that, we can say the antibody neutralizes this pathogen and causing this pathogen no longer able to injure or to damage our cell. Well, agglutination means that the antibody will bind to the antigen on this pathogen and cause all of these pathogens to clump together. So when all of these pathogen or the antigen are clumped together, then they will form a very big complex, and all of these pathogens will no longer able to move. So with that, yes, it actually caused these pathogen to form a huge complex and no longer able to attack our body cell. Well, lastly, precipitation. Precipitation is normally for those soluble toxin. Those soluble toxin that is very hard to be cleaned up by the phagocytosis will be bind with the antibody. So this antibody will bind to this soluble toxin and form a way bigger complex. So this bigger complex will make this toxin to become insoluble. So when it is insoluble and precipitate out, then this toxin will no longer able to cause any damage to our cell. So the outcome of this action of the IgG, IgM, or IgA is going to form bigger complex. And this bigger complex will later be engulfed by the phagocytes easily. Then next, we will move on to the outcome number 4, which is the activation of the antibody-dependent cellular cytotoxicity, ADCC. Antibodies like the IgG or IgE will bind to the antigen that occurs on the either the pathogen or it can be the infected cell. So for example, a virus infected cell. So virus infected cell will have the viral antigen on their surface. So the antibody like IgG will go and bind to this antigen on the surface. So after the antibody bind to the antigen using the FAB region, their FC region will bind to the FC receptor of the effector cell like the natural killer cell here. So when these antibody that bind to the antigen bind to this natural killer cell via their FC region, it is going to activate this natural killer cell and cause the natural killer cell to release the factor like perforin to lyse this virus infected cell. So with that, the antibody eliminate those viruses that occurs in this infected cell and thus give the protection to our body. So the same thing can occur if, let's say, it is a parasite. So same thing, the antibody can coat the parasite and then use the FC region to bind to the FC receptor on the eosinophil and thus activate the eosinophil to kill the parasite. So normally, the antibody that is going to activate the natural killer cell to kill those cancer cell and also the virus infected cell would be the IgG. While the antibody class that is going to activate the eosinophil and thus kill the parasite or allergic and causing allergic reaction would be the IgE. Next, we will look at the mast cell degranulation. The antibody will sometimes coat the mast cell, and this class of the antibody is normally the IgE. So when the IgE coat the mast cell, this process will be called as the sensitization. So when this mast cell encounter any antigen that can be recognized by this antibody on their surface, then these antigen will bind to the antibody. The binding of the antigen to the antibody on the surface of the mast cell is going to activate the mast cell and thus causing the degranulation of the mast cell. So mast cell is going to release a lot of the substance like histamine. And this histamine will later cause the inflammation and also the allergic reaction. Lastly, we will look at the outcome number 6, complement activation. The antibody can bind to the antigen and form a complex called antigen-antibody complex. So this antigen antibody complex can activate, it can act as a complex to activate the first complement protein. And the activation of the first complement protein will activate the second complement protein and then the third complement protein and so on. So this will cause the activation of the cascade of the complement protein. So all of these complement protein will later act together to form the membrane attack complex MAC, which will create the pores on the cell and cause the lysis of the cell. At the same time, some of these complement protein will also cause the inflammation. Normally, the class of the antibody that will cause this outcome would be the IgM and also IgG. IgM is a strongest class that can cause the complement activation, while IgG is less effective.